Hello. Today I'm covering the circulatory system. So as part of the cardiovascular system, we not only talk about the heart and everything that's happening there, but we also have to understand the circulatory system. So all of these vessels that are taking the blood around the body, taking oxygenated blood to the tissues, bringing this deoxygenated blood back to the heart. These vessels have a very important role in the cardiovascular system, and that's what this chapter is about, is just concentrating on these vessels. And it's a good to cover this particular chapter before we discuss blood pressure regulation, as we will be talking about certain characteristics of vessels that are implicated in blood pressure regulation. So you'll understand more when I get to blood pressure regulation. So this is a good chapter to cover before that. Here are the instructional objectives. I noticed that the last two bullets really belong to that next lecture. So I apologize for still having them here. But they will be part of the next lecture about venous return functions and blood flow velocity and blood pressure and all of that. Okay, so the circulatory system is about blood vessels and the categories of blood vessels, and we're going to talk about arteries, veins, and the capillary beds that basically connect those two. Arteries are considered efferent vessels. They are vessels that are coming out of the heart with the oxygenated blood, except for the pulmonary arteries, right? So right there at the heart, we had a very specific terminology. If you remember, I discussed that in the heart anatomy chapter. The arteries at the heart, they don't all carry oxygenated blood. We also have the pulmonary arteries that are taking deoxygenated blood to the lungs. But in the rest of the organism, in the rest of the human body, arteries are described as being the efferent vessels going out there to deliver this oxygenated blood to all the tissues and delivering this oxygen and nutrients to the cells. And the cells at the capillary beds are exchanging this oxygen and these nutrients with carbon dioxide and waste products from their metabolisms or whatever else. So that's exchanged at the capillary beds, and the blood becomes deoxygenated and carrying waste. So via the veins, and that's why they're usually illustrated as blue, and we describe them as being the afferent vessels. So the capillary beds are very, very microscopic vessels that connect the smallest arteries to the smallest veins. So we have these two systems that are connected at the capillary beds where that exchange is going to happen. Okay. A quick illustration here. This I'm surprised it's not actually in the legend. This is most probably, it looks like it to me, a scanning electron microscopy picture of a vein over here and an artery over here and all of these little capillary vessels in between. So in every tissue, you will have all of that. You will have the arteries coming, the veins leaving, and all of these little capillaries that are trying to be this the connecting vessels. So we focus first on, let me reduce this in size so you can see the picture even better. So we're going to focus on the anatomical structure of these vessels, and we're going to talk about the vessel walls. And the walls of these vessels is made of 3 layers that we call tunics. And we use the word tunica adventitia, tunica media, and tunica intima. Intima being the innermost, media in the middle, and adventitia the outermost. So tunica adventitia over here, you can see it's this outer layer, and tunica media is the one that includes the smooth muscle right here. And tunica intima is basically this whole section over here, this. So these layers over here make up tunica intima. So each tunic actually is a combination of layers of tissues. So that's what we're going to see one by one. This is a really nice diagram because it just shows you this layering, and it pulls the inner layers kind of forward. That way you can, see the layering. All right, so smooth muscle in tunica media. What else did I want to say here? There's an elastic membrane in between, an elastic membrane even on the outer surface here. Lamina propria is usually a term that we use at about the connective tissue at the base of epithelia in a mucous membrane, for example. We call that connective tissue the lamina propria. In this particular situation here, the lamina propria is not just the connective tissue, but there's actually a smooth muscle layer. So if I wanted to use red here for muscle, this would be it. So this is kind of muscle and connective tissue. Then you have the basement membrane, very, very thin layer of kind of bonding cells, bonding molecules. These are secreted, not cells, molecules secreted by the cells above and the cells below, and they create this kind of bonding layer between the other two tissues that are on both sides. So on the innermost side, there is that finally the endothelium, which is the simple squamous epithelium of blood vessels. Simple squamous epithelium. That's what endothelium is. It's the same thing. It's a simple squamous epithelium. The cells are very flat, fusiform, and only in one layer. And these cells contribute to the basement membrane, and the cells from above will contribute to the basement membrane, like this and like this. And that basement membrane is between those two layers. Okay. So this is the general layout, right? These tunics, tunica intima, media, and adventitia, with kind of a general indication of what we would see in these different tunics, with the intima being the innermost layer. So that's where you would logically see the epithelium, because the epithelium lines the cavities. That's the definition of epithelium. So that's where you see your epithelium, and then all of these layers with the muscle, smooth muscle, being in the tunica intima. There's more smooth muscle in media, and then the adventitia. So for each vessel, there will be a slightly different variation of all of that. So here we go. If we're looking at a large artery, conducting artery, we call them the large and kind of moving blood through the body. Of course, the lumen is that opening. That's what lumen means. And what's the first layer? Is tunica interna. And what does it have? It has the epithelium. That's the lining of that inner cavity. This lining over here is the simple squamous epithelium that we expect at lining the lumen of vessels. And then we have a basement membrane, just like we saw in the previous slide. And then we have tunica media. And in tunica media, there is a lot of elastic fibers, in tunica media. In tunica media, where we have smooth muscle, if we go back here, I want to show that to you. Tunica media, smooth muscle. See that? That's what I was. That's what I highlighted earlier. Smooth muscle around. So this is basically your thicker smooth muscle right here. That's where the main smooth muscle layering is in tunica media. We have a little bit of smooth muscle here, but when we talk about vasoconstriction of the vessels, we're talking about that smooth, thicker smooth muscle layer in tunica media. So in that thick tunica media muscle layer, we have elastic fibers. And that's different from what we've learned so far. We've never seen elastic fibers dispersed in the smooth muscle, but that's the way the arrangement is here. We have smooth muscle cells, and in between, we have elastic fibers dispersed throughout. So that's very important because elastic fibers allows for elasticity, gives the vessel its elasticity, and the property of elasticity is the ability to recoil after it was stretched. So it's not just about stretching. Elasticity is, stretch alone is not good enough. To be able to be considered elastic, you have to be able to recoil. And of course, these large arteries, we want them to recoil every time they pump up with blood, and then, the blood gets moved forward, then the vessel goes back down in its diameter, etc., over and over and over. So elasticity is very important in tunica media. And then we see a tunica externa with that gets pierced with what we call vasa vasorum. It's the capillaries that literally go feed the cells of the vessels. So you have these tiny capillaries that go back into the vessels, because these vessels are made of tissues that are made of cells that also need to be provided oxygen and nutrients. A medium artery will have a slightly different layout, and this is where you see the internal elastic lamina and external elastic lamina. That's just a different layering order. You still have your smooth muscle in tunica media, except that you have a couple of extra laminar elastic layers. Let me see if I have anything else to say about medium arteries. No, that's what I want to say. Okay, so we get into, now we're getting into smaller vessels, right? Large arteries, medium arteries, and now we're into arterioles. Arterioles have a tunica interna, of course, endothelium, basement membrane, and a smooth muscle layer that is not as thick as previously, right? In tunica media, we still have that smooth muscle layer around, and tunica externa. So much more straightforward layering in an arteriole. And then finally, in the capillary, we really don't want all of this layering, because at the level of the capillary, this is where we're going to have exchange of gases, with O2 coming into the tissue after leaving the vessel. If it's a oxygenated blood, it leaves the lumen of the vessel and diffuses according to its partial pressure gradient, and because that's a gas, and the gas is going to move through the epithelial tissue, so to get into the cells in the surrounding tissues, So we definitely want less layering. We want this process to be very efficient. So the basement membrane is always going to be there. So here we have a simple squamous epithelium. Oops, it goes in. And same thing for CO2, it will also diffuse into the in and out, just like the oxygen molecule. All right. So this is where we are. This is the summary on the right side of the structures of the three types of vessels we saw, and the capillaries at the bottom. So we already talked about the arteries, the large arteries, medium arteries, and arterioles. And these are going to come to reach the size of very small capillaries that form the capillary beds. And it's at the capillary beds that we're going to have this big exchange of gases and nutrients, and the blood keeps flowing. And as it keeps flowing, it gets into the venous system, and it starts by being in venules that turns into medium veins and then large veins. And the structures are going to be a little bit different as well, just like we had similarly seen that for arteries. We're going to see that for veins. Yeah. Okay. So before I get into those veins, I want to say one more thing about arteries. You have to be aware that there are arterial sense organs in very specific locations on the arteries very near to the heart. So I wanted to show that to you here because we're talking about arteries. And so this is actually quite important. And we will mention those again when we talk about blood pressure regulation. So what are arterial sense organs? It's these things that are colored here in green, more teal, or green. So let's talk about the structures that are in teal color. I'll just use a blue. So one is called the carotid body. This is the carotid body over here. And the other one are the aortic bodies over here, these two. And both of these are chemoreceptors. Chemoreceptors. One is on the external carotid artery, which is what this is. And that's a branch of the right common carotid artery, which we now know is a branch of the brachiocephalic trunk here. And the aortic bodies are directly against the aorta. Yes, on the aortic arch. And these are chemoreceptors. They are going to be very important in sensing the concentration of ions in the blood to let the brain know if there are too many hydrogen ions, the blood pH is dropping too low, and we need to regulate that. Or whatever other ions are passing by, they are being monitored by these chemoreceptors. So very important to know that they're there on these arteries. And then we have, I'll do it in green, we have baroreceptors at the carotid sinus, right here. And this one is on the internal carotid artery, the carotid sinus. And we have these baroreceptors. Oops. Let's stay consistent with what I've been doing. Just underlining baroreceptors right here on the aortic arch. I'm just trying to stay consistent with my writing. Carotid sinus, aortic arch, both have baroreceptors. Carotid body and aortic bodies have chemoreceptors. Chemoreceptors. Okay, so what are baroreceptors? Baroreceptors sense the change in pressure. Oh, this is going to be so important because the pressure, look at all of these baroreceptors over here. The pressure right here at the base of the aorta and in the aortic arch, the pressure in that portion of the aorta is very important. It needs to be elevated enough that the oxygenated blood goes far enough. If blood pressure is very low, we're going to have issues with providing the oxygen necessary for the brain to function, and of course, you know, the other parts of the body. So we'll talk about these again in an upcoming lecture. So this is a summary of the features of arterial structure, and I wanted to kind of summarize them on this one slide before I got into veins. We call them resistance vessels because they are built to withstand pressure surges. Pressure surges, and they have this elasticity in them, especially in the large vessel. There's this elasticity, so it allows for the surges in pressure. Pressure meaning pressure comes because of the volume of blood in the vessel. When volume increases, there's more pressure against the wall of that vessel. So that's what we're talking about when we're talking about pressure. So they're built to withstand these kinds of pressure. They're more muscular than veins. The muscle layers are thicker. So that gives them a little bit of rigidity. They can retain their shape even when they're empty. And if you noticed at a cadaver lab dissection, if you had the opportunity to catch that, the aorta was huge and wide open, and it was like a pipe sitting right there in the body. It was really fascinating to look at, as opposed to other smaller vessels are kind of collapsed, and it's really hard to see. Okay, arterioles have thicker tunica media in proportion to the lumen diameter, so that's kind of one characteristic of arterioles. Meta-arterioles, which are, you know, a bit smaller than arterioles, but not quite capillaries yet. Meta means ahead of. They're short vessels that link the arterioles to the capillaries, right? So they're ahead of the arterioles, right? Meta means ahead, like metacarpal, ahead, but not quite capillaries. So they're kind of like in between. They have individual muscle cells forming capillary sphincters. This is really cool. I'm going to show you a couple slides about that coming up. Capillary sphincters. Okay, so this is really important for basically controlling where the blood is going to go. When I show you the picture, it's really going to make more sense. And this will be much more useful instead of having a continuous tunica media. And you'll see what I mean by that, by having these individual muscle cells there as pre-capillary sphincters. You'll see why it's important. Okay, and then the summary of these arterial sense organs: baroreceptors of the carotid sinuses, they respond to changing blood pressure, and the chemoreceptors of the carotid bodies and the aortic arch, they respond to changes in pH. So the example I had given you was the levels of hydrogen ions, and that's linked to pH, but also the levels of CO2 and O2, the gases. So let's talk about these precapillary sphincters that I just mentioned to you for the first time. So we have in these meta-arterioles, we have smooth muscle sphincters. So sphincter muscles that are basically controlling the flow of blood, whether it goes straight through this the arterial and into the capillaries or not. So they are sphincter muscles that are basically controlling the flow of blood, whether it goes through or it doesn't go through. So these sphincter muscles are much better at doing that than if we had them throughout the tunica media, right? Instead of having them all throughout the tunica media, then that means it would be all along the wall of the blood vessel. But here it's actually at the base of each branching, you know, when you're going to have this capillary bed with all of these branches of capillaries, you're going to have these sphincter muscles that are literally squeezing the base of these branches and preventing blood from going through. So the example here is shows you the smooth muscle here of the precapillary sphincter contracting, and when it contracts, it prevents the blood from going through as it did before, and instead it's just going to continue with this flow over here. And this is going to be really important, and we will cover that. Well, actually, yes. So I'm showing that to you over here, and then at the end there's also one more slide that will bring this back up. So look at here. We have the example of sphincters being, sphincters being open. And then I'm going to show you the next slide, they're closed. So if the sphincters are open, it allows this blood to go into the capillary beds. This is what this is. These are all the capillaries. That's the capillary bed. Right? That's what we would call a capillary bed. This whole thing. So the blood flows and actually is not prevented from going into these capillaries. So we're going from arterial to meta-arterial, right? This one here. This is the meta-arterial, and this is controlling what gets into the capillaries. So the meta-arterials are after the arterioles, and here these small meta-arterioles are those branches that have these smooth muscles around their bases, and basically either being relaxed and allowing the blood to flow through the capillary bed, perfect, or not. So here you have these sphincter muscles completely constricted, contracted, constricting the meta-arterial and not allowing the blood to flow through the capillary beds. So no flow here, no blood flow here, because the blood is going straight, basically through, and just providing, just basically quickly dumping oxygen and taking whatever it can as far as carbon dioxide, and getting into the venous circulation. So we have, so that's how blood gets recirculated. It can go into the capillary beds, or if we don't need it to go in the capillary beds, these sphincter muscles are going to contract to constrict the meta-arterioles. So one of the last slides, we'll explain the scenarios of when that happens. So looking at these capillaries, let's see how they're made. So you have endothelial cells, which are your yellow cells. So this is an endothelial cell over here. There's another one over here, except that you don't catch its nucleus, and another one over here. But again, you don't see its nucleus, but the one over here, we happen to catch its nucleus. So when I say catch, it's only because when you slice the tissue, you might go through exactly in the plane where the nucleus is going to be, but maybe it's kind of off in the cell, and your microtome is going through the cell exactly right next to the nucleus, and then you would never see it from the cut, but it's there. It's just kind of deeper. So around these vessels, we have, supporting vessels called pericytes, the basal lamina at the base of the endothelium. We have already seen that. We have intercellular clefts, which are opening, and we'll see that these openings actually vary in width. What else? Most of the time, we have tight junctions between these cells because there's no business for the blood to just be seeping through for no reason unless we are right there in the capillary bed. And we're going to see, how does this exchange take place. So here we go. The types of capillaries. Either we have continuous capillaries, which are connected with tight junctions, and basically there is no escaping from the vessel itself because all these endothelial cells are tight together. Or we have what's called a fenestration, and we call this a fenestrated capillary. And these are the gaps. Fenestra with diaphragm. So the diaphragm is like like the thinnest ever possible little membrane, but that's going to actually, in cases where we actually need the passages of particles, we would not see this membrane. So this is an example of fenestra without a diaphragm. So maybe you know a little bit of French. So actually, the word fenêtre, une fenêtre, means a window in French. So this is a fenestration. It means these holes that you can actually see through, and you can see through, so you can go through, and that's what this fenestration is important for. In some cases, we have such huge openings that these are called large fenestra. So we describe these as being sinusoidal capillaries. So sinusoidal capillaries are the gaps are much bigger than the fenestrations would be in a fenestrated capillary. So we call it sinusoidal. It's like a sinus. It's like that big. It's bigger, right? A sinus is an opening, and it's definitely bigger than a fenestration. Yes. So actually, you will see an example of sinusoidal capillaries in kidneys, in fenestrated capillaries in kidneys. All right. So, so here's a diagram of an endothelium with an endothelial cell for which we can see a nucleus for this one, no nucleus for these in the view of the plane of view, but it would be, a little bit like right behind probably, if we kind of continued our sections. So this particular diagram is showing fenestrations. So where you have openings, and that allows for things from the blood to, go through. Otherwise, you can't just go through unless you're a gas and you can diffuse. So under a scanning electron microscopy with the capability of scanning electron microscopy, you can literally see these fenestrations. You can see them as these little circles. This is the visual of what they look like on the surface of the endothelium. Because you just saw a cross section over here in the previous slide. You just saw a cross section, and then you see, oh yes, there's a gap here, and there's another gap here. But we're talking about holes on the whole surface, right? So if I drew this in 3D, let's look at 3D. Yeah, this is 3D, right? This is the vessel, the tube, right? So we actually have holes in the vessel, and you can see the hole sideways over here. See these holes? That's a hole. It's actually a circle that you can see throughout the surface of this endothelium. Here you're looking at it sideways, but it's all over the surface. So in a microscope, you actually see that. You can see patches of fenestration next to areas where there's no fenestrations. So this is just basically the two pictures I already showed you that are just placed side by side. So what's so special about capillaries? There's a little bit of kind of like reviewing here. They have a diameter smaller than a red blood cell. So very, very, very small. And red blood cells can go through because they can bend. And that's the problem in sickle cell anemia. Those red blood cells have a hard time bending, so they can't get through the capillaries. So the average diameter is 5 micrometer at the proximal end. So the proximal end, so that would be like the beginning of the vessel until it's, before it gets all the way to the end of the capillary. And a red blood cell on average is, between 7 and 8 micrometers. So it's bigger. It's bigger and it will bend. So we have 1 billion capillaries with a total surface of 6,300 square meters. I don't know if that speaks to you. If it does, good. If it doesn't, it's not a number I would ask you to memorize. But we have bazillion. That's not a word. We have 1 billion capillaries because we have so many places we need to feed. So we have to have all these capillaries everywhere. So the type of capillaries, that's what we saw: continuous, fenestrated, or literally discontinuous. We call them sinusoids. Continuous, the endothelial cells are held together with very tight junctions, and endothelial cells are separated by narrow intercellular clefts. I showed that to you on the diagram. Brain vessels lack intercellular clefts. We have what's called the blood-brain barrier in the brain. This is a completely different setup for brain. We don't just use tight junctions. We also have, I don't know if you remember, we also have astrocytes. These are glial cells in the brain that form perivascular feet around these tiny blood vessels. And basically the astrocytes are hugging these vessels and controlling what's going in and out, if anything. So that that's what's called the barrier in the brain between the blood and the brain tissue. The brain-blood barrier is made with the astrocytes around these capillaries. So fenestrated vessels, they're riddled with holes. We saw the beautiful pictures of the microscope. This is important in organs that engage in rapid absorption and filtration. So we'll see them in the kidneys. I had mentioned that to you. Small intestine, in the endocrine glands, because of that, movement, rapid movement of things in and out. And then the sinusoid ones have these very wide gaps. That's what kind of a sinus is referring to. It's really bigger than just a fenestration. No basal lamina, very large openings. This is when proteins and red blood cells literally can get through and out of the vessel. So we'll see those again in kidneys. You'll see. All right. So all of these are organized into capillary beds in the tissues. There's a network of 10 to 100 capillaries that can be supplied by single meta-arteriole, and we saw that in this picture over here, right? So this entire capillary bed, let's say all of here fed by this meta-arteriole, and so we have a lot of these. These are all the capillaries are in networks, and these networks are fed by the meta-arterioles. And all this is regulated by these precapillary sphincters that are squeezing and constricting the blood flow to the capillary bed. So look at this. We have these capillary beds pretty much everywhere. There's a couple different layouts that you could kind of be familiar with. Simplest pathway is one capillary bed. We go through the bed, we dump our stuff, good stuff, and then we take on the CO2 waste and kind of leave and go out, right? So this is it. Coming in, going out, and going through, of course, through all of this, the capillary bed. Sometimes there's something what's called a portal system. A portal system, actually, there are two capillary beds that are continuous. So you'll have some of the exchange in the first capillary bed and the remaining of the exchange in the second capillary bed. And, and then the blood will come out of this and finally be sent to the venous circulation. Arteriovenous anastomosis. This is what's called a shunt right here. This, for example, if a capillary bed is not available, if it's for whatever reason, there is such thing as shunts that could be there to connect, an artery with a vein and still distribute some blood to the area. And of course, all of these vessels, as much as we kind of draw them as simple as this and that first one, it's not that simple. These arterioles, or all of these vessels, they anastomose. Anastomose means they fuse together and then they will branch out and anastomose again and branch out. It's like the plexuses with the spinal nerves. So anastomose means they fuse and then they branch out, creating new branches and fuse again, anastomose and branch out. So that's what that means. All right, so all this was about arteries. So I'm going to get into veins. Maybe not spend as much time because it's just about learning the layering of all of these and the differences between them. So we have a tunica interna with an endothelium. So this is pretty much the same. Tunica media has a narrower, smooth muscle layer, narrower than the arteriole. Remember, I mentioned that earlier. And then we still have a tunica externa. So it's in the medium veins that we finally see valves. So what are valves? That's really cool. In those veins, we have these little flaps. It's exactly like, well, in concept, it's like the valves in the heart. These valves are kind of like swiveling doors that kind of swivel out of the way and then shut back, and swivel out and shut back. But just like the valves in the heart, this is to prevent backflow. So they really only open in one direction. These valves are going to open like this. And when do they open like this? When the blood flows through them. So when the blood flows through, it pushes these valves to open sideways. And then when the pressure drops back down, the blood pulls backwards and kind of plops into these cups and helps shut the valves back together. So a little bit like at the base of the aorta, where you had these semilunar valves, the aortic semilunar valve, that are kind of cups. And when the blood falls into these cups backwards, after the initial ejection, when the blood pressure drops in the ventricle, right, the blood kind of goes backwards just a tiny bit, and it kind of shuts those semilunar valves. It's the same concept here. These valves are designed, they are constructed in a way where the flow of blood is only in one direction. Of course, that's if the valve is working properly. If it's not working properly, the blood is not going to kind of move forward, and it would be pulling at the in your lower limbs, because that's kind of where gravity takes you. Okay. So in large veins, so just, I mean, I discussed this, and when I discussed, I believe it was blood flow through the heart. You know, how is the blood coming into the right atrium via the superior inferior vena cava? How is the blood arriving there? That's because it's being, you know, milked up there, upwards. And I mentioned that in a previous lecture, where you have the contractions of the skeletal muscles in your leg, because when you're moving, I mean, literally anything you're doing, like you're just bending your knees under the table, that's a movement. Muscle contraction takes place. These muscles, when they contract, they're squeezing those veins that are running kind of through them. And the whole purpose, well, it's kind of like a secondary effect. The muscle is contracting because you're moving your leg, walking. So the muscle contracts, but at the same time, when it contracts, it actually squeezes these veins. So it's kind of, it's like pushing the blood upwards. We need that because gravity is in the opposite direction, and we need to bring the blood upwards, back up to the heart. So skeletal muscles do that squeezing. The second thing is the milking effect, which is kind of like it's kind of like this process with the involvement of smooth muscles. It can also be smooth muscles kind of contracting to move the blood upwards. So it's kind of like a milking effect, you know, flip, 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 like this, and you have the blood that's going to keep going upwards, upwards, upwards. And also, I do remember saying that when you breathe in, every time you inhale, your make sure I don't have this backwards. When you inhale, your thoracic volume expands. When volume expands, pressure drops. It's Boyle's law. It's inversely proportional. When the volume decreases, pressure rises. When the volume increases, pressure drops. So when you're breathing, your lungs are opening wide open, right? The rib cage is being kind of pulled apart by all these respiratory muscles. That volume increases horizontally and even vertically because of the diaphragm. And that means the pressure drops. And when the pressure drops, it pulls the gases in. So that's how you breathe. So what your thoracic pressure drops, that means the abdominal pressure goes up. And when the abdominal pressure goes up, it actually kind of like puts that pressure on these large veins going upwards to the heart. And yes, so every time you take a breath, you're actually increasing your venous return. That's where I was getting at. You're increasing your venous return. Every time you do that, there's kind of like a squeezing of the blood upwards into the right atrium. There's blood going into the right side of your heart. And if you do it a lot of times, you'll start getting lightheaded because what I want to say is your heart rate will go up. Because every time you're doing that, you're basically dumping blood into the right atrium. So the heart's going to pump it. And then, you're doing this over and over a little bit, kind of like frequently. If you're doing it over and over, you're basically kind of telling your heart, okay, do it, pump it, pump it, pump it. And your heart rate will go up if you measure it. Okay. Large veins have these larger smooth muscle layers. So that's, part of the milking action. So the features of these venous structures, they're more described as being capacitance vessels, where they have a thinner wall. They're definitely flaccid. When they're empty, they kind of plop down. But, of course, they're expandable. That's, they have to be there. Going to get filled up with blood, and then the pressure is going to go up and like this. But the pressure is not high. It's a relatively low blood pressure, non-pulsatile, because it's not driven by the heart pumping. The heart pumping just kind of took out the oxygenated blood out of the aorta. And that's where all the pressure was. By the time you pass the capillary beds and now you're in the venous system, there's really not much pressure. And now we just kind of have to work this blood back upwards with the muscles, with the breathing, and all the pressure inside your body to kind of like get this blood upwards. That's how it works. So from smallest to largest, we call them post-capillary venules. So those are the ones receiving that deoxygenated blood from the capillaries. Actually, it's basically one continuous vessel. But when it's a capillary, it's oxygenated and then it's deoxygenated, but it's the same vessel. Yes, so at that level, we don't need those muscles there. In the capillaries, it's just about being a capillary, a very thin vessel, just for the exchange of these gases. At the level of the venules, we have a more porous than capillaries, because that's where most of the exchange of fluids takes place, as opposed to the gases that were exchanged previously by the capillaries. And so because they're more porous, this is a perfect spot for these leukocytes, the white blood cells, to kind of leave the bloodstream and get into the tissue, and do their thing if it's needed there. The venules have a tunica media with 1 to 2 layers of smooth muscles. So that's starting to build up. medium veins. So we have the same setup, tunica media, relatively thin with smooth muscles. This is not a surprise. This is where we have seen the smooth muscle being in the tunica media. I'm keeping repeating myself here. And the tunica externa is relatively thick. All right. And this is how we actually have the valves formed, is the infoldings of the tunica interna. So basically it's a continuation of the tunica interna instead of just being kind of flat in very specific spots. We actually have this invagination of the tunica interna to create these valves. Okay, so in large veins, the smooth muscles are in all the three tunics, because we really need those muscles to move that blood upwards. And the venous sinuses have these very large lumen, no smooth muscles. For example, coronary sinuses of the heart, the dural sinuses of the brain. We're talking about really large spaces. That's what we're talking about. So a good example of that is the dural sinus that runs in your brain. It's basically the visceral layer of the dura mater. I'm like trying to concentrate and find my words here. So the dura mater around the brain has actually two layers, a parietal layer on the outside, and the visceral layer on the inside actually dives down between the hemispheres and creates this kind of like partition wall. I'm not sure if you can visualize that in your head, but we'll see that with the brain chapter. And it creates these kind of like connective tissue partitions between the hemispheres. But as it dives down the visceral layer of the dura mater, it separates from the parietal layer, and you have that gap, and that's filled with that's the venous sinus. It's filled with that venous blood, and that's basically where the CSF gets recycled in this venous sinus, and it goes into the venous circulation via the arachnoid villi. I don't know if you remember that, the arachnoid villi that kind of pierce through the dura and dump the CSF into that sinus. So that's pretty cool. But I didn't think I was going to talk about that extensively here, so I don't have a diagram to show you, but that's more part of the brain anyway. Okay. All right. So that's what we did. We went from a large artery to medium artery to arterial, and these change into capillaries. The capillaries form basically a capillary bed where the oxygen gets delivered and CO2 gets picked up, and now we are into the venous circulation. It starts with venals that will become medium veins with the valves, and to much larger veins that are bringing this deoxygenated blood to the heart. And of course, they come from the head and neck, as well as lower limb and lower parts of the body. Okay, so we're pretty much towards the end. There's only 4 more slides left. So this next slide is to actually show you these venous valves. Maybe I could have inserted that slide a little bit sooner, no big deal. But so this is pretty cool stuff here. Look at this. I'm gonna use blue for deoxygenated blood. So here it is. When the skeletal muscles of the muscles in your legs, for example, contract, when they contract, they actually kind of squeeze the veins that are going between them, through them, and it allows to squeeze this blood upwards, right, like that. But when they relax, when these skeletal muscles relax, what actually is happening is that you're decreasing that pressure on this vessel. So the blood kind of goes backwards. It falls into these cup-like shapes of these valves, and actually this helps close the valves back together. Like here, like this, and the valves kind of close back together. It's pretty cool. Okay, so this is going to be really important when we talk about velocity of blood. I'm going to mention this back in the next lecture when I talk about that, and the resistance in the arteries depending on the diameters. So I'm mentioning it here, but I'm going to bring it back up in the next lecture. So we go from very large arteries to arterioles, blah, 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 and then we end up in the capillaries, and all of a sudden the velocity decreases. So if you're looking at here, this is velocity of blood, which is kind of like the speed of blood, right? Milliliters per second. Boom, 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 boom. And then all of a sudden it arrives. This is the area where you have the capillary bed. This is the beginning of the capillary bed. Boom, velocity decreases. The blood literally slows down in the capillaries, slows down in the capillaries. You have the time to exchange all the good stuff and get rid of all the bad stuff. And once it's in the venals, then we can kind of pick up on speed and get back into the larger veins. So how come we have velocity decreasing? Well, we can understand, why. So physically, how come we're going to see that? But the why, as far as, why do we need to do that? And then there's why, how come this is happening physically, this coming up here. But why do we need to do that? Because we need to distribute this blood to all of the tissues that are surrounding this capillary bed. And we need to make sure there's time for the oxygen to get delivered, the carbon dioxide to come through, the nutrients to kind of get dumped, and the diffusion of all the waste to get back into the blood. It takes time, so we can't just rush through it. We have to let the blood kind of like, it's like the lazy river at this point. We're just kind of like sitting in our tube, and we're just kind of going down the lazy river and just making sure that, hey, you want this? Hey, you want that? And we're kind of like distributing things to the tissues and picking up, hey, you don't want this anymore? Let me take it. And, so the velocity drops. So how can it drop? It drops because the diameter of the vessels are decreasing. So the blood has traveled a really great distance at that point. So let's imagine this is a capillary bed in your foot. But we're talking about velocity right out of the aorta, and then by the time it gets to your foot, okay, velocity decreased just because we're far away. So that's the first point here. Blood has traveled a greater distance, so, it's slowing down. And along the way, it just, from all the resistance along the way, it just kind of slowed down because of all this tubing. And that's every time you touch something, that's slowing you down, the walls of the tubes. So not only that, the radius of these tubes is diminishing. We started with this big artery, and then now we're into a much smaller vessel. When the radius goes down, it actually increases the resistance. And I'm going to review that again in the next lecture. It increases the resistance. How is that possible? Well, if there's less space, because now we're in a smaller tube, we're in a small tunnel, and we're the same number of people that were like all leisurely in this huge tunnel, just walking far apart. Hey, I see you over there, I'm over here. But now everybody has to walk through the tiny tunnel. Now you're all against each other and against the wall, and right. So this is like slowing you down. There's just too many, I say people or cars going through a tunnel, right? When you have all of these cars coming from 5 different lanes into, well, this is a better example. All these cars coming from 5 different lanes into one lane, and they're gonna slow down, and then one car at a time going through that one lane, right? So that's when the diameter decreases, the resistance increases. So it's harder to get through. That's what it means, the resistance. It's harder to get through. So we have this the small radii, we say in the plural of radius. However, the way we look at this in a capillary bed, we actually look at the sum of all of these radii. So despite the smaller radii, you look at the total cross-sectional area. So the total cross-sectional area actually increased because you're looking at this cross-section plus this cross-section, this and this and this and this and this and this. And when you add them all up, it's much bigger area. So that has the effect to slow the flow of blood. Okay. So on the other end, you have the capillaries that are going to get into, eventually, we need to go all the way to get to the inferior vena cava or superior vena cava, but the vena cava to the heart. And velocity needs to rise because now it's about taking that blood back to the heart. So veins in general have larger diameters, not in general. The veins, they go from being, venules and then medium veins and large veins. So we're getting from small diameter, large diameter, large diameter. So now you have decreased resistance, right? The walls are so far apart, there's less resistance. We can flow better. So that helps with velocity. We go a little bit faster when there's less resistance. So progressively smaller channel increases the flow rate, right? So when you actually have the flow rate was decreased here, well, we saw it over here. It was decreased here, and then we re-increase it over here because now we only have we have a bigger channel and not as many to go through, but only one. So here, just to simplify things, only one. Okay, so I'm going to revisit that in the next lecture because it's going to be important for blood pressure and that whole discussion. Okay, so the last 2 slides have to do about this local control of blood flow in the tissue. I want to show you how important this is. So I told you earlier, arterioles, pass through with this blood, oxygenated blood, great. And you have meta-arterioles over here, these here, that are basically your vessels that are between the arterioles and the capillaries in the capillary beds. So at the level of the meta-arterioles, we have these sphincter muscles. These are smooth muscles that are specifically circling the base of these meta-arterioles and controlling what's going to go from the arteriole to the capillary bed. Are you going to be allowed to go through and get into the entire capillary bed? Or you're going to be asked to just forget about the capillary bed, keep going, nothing to see here, just keep flowing, right? So that's going to happen. And it's going to depend on what are you doing. And that's what we talk about when we say rerouting of the blood flow. This is so interesting because depending on what your body's doing, we need the blood to kind of supply the cells that are the hardest at work. So if you are, playing soccer on the field, running, whatever it is, your skeletal muscles in your legs are really working hard. You want to make sure you're sending that blood to these muscles. They need to be vascularized and provided with the oxygen they need. So you have this blood flow that comes down from the aorta, and instead of allowing this blood flow to go to your digestive system, which would be out of the superior mesenteric artery. We saw the mesenteric artery is one of those arteries, kind of like branches off of the abdominal aorta. So that provides, mesenteric arteries are providing the blood to the digestive system. We're not worried about digestion over here. We need to make sure the blood goes to the muscles in your legs. So constricted, reduced flow to the intestines. That's what's going to happen. So that's where you're going to see these sphincter muscles contracting and constricting the flow of blood to the intestines, because we need the blood to go down to the legs, and we need the increased flow to the legs. So if we are relaxed and we're not running, we're not doing anything, our feet are up, they're not even moving, we just had a nice good meal, well, this is when the parasympathetic nervous system is at its best, where you really have, no other worries than to just relax and let your digestive system work. So in a situation where you are just you are just relaxing, we will have constriction to reduce flow to your legs because you're not really using them, and we need the sphincter muscles to be dilated at the level of the superior mesenteric to allow an increased flow to the intestines, because now you need that blood there, because that's what's happening when you're just relaxed. Your parasympathetic is handling digestion, and we need to make sure that all of these cells of the digestive system are provided with all the nutrients and oxygen they need. So these are a couple diagrams, pie charts that illustrate at rest versus moderate exercise, total cardiac output. Of course, when you're exercising, it's much higher cardiac output, 17.5 liter per minute. This is regular cardiac output. It's usually just 5 liters per minute when you're just chill. And where is that 5 liter going? So 20% to their muscles, because of course you still need to provide blood to your muscles. About 27% goes to the digestive system. Your kidneys, they never take a break. 22% of them go through the kidneys, this blood, and your brain, 14%. And I don't need to go into the rest. But when you are exercising, a lot of this blood is going to your muscles, because that's what's really working the hardest at that point. Okay, I think this was a great, well, look at me saying this was a great lecture. Hopefully you will think it's a great lecture. It was a great lecture on vessels, arteries, veins, their structures, their functions, and how they are important for the whole body depending on our activity. And that's it. That's it for today. Thanks for your attention. That's it. Bye-bye.